In the first part of the demo, I'm going to show you the different tables of the selected database, the layout of different viewing and formatting options of IJC, and will import tables into Excel. In the second bit, I'm going to show you the different searching options for Markush structure in Instant JChem. And finally, I will demonstrate some of the enumerations which you can use to generate your own Markush library. First, have a look what we have in our local database, and then how we can view the content. In the left panel, the database content can be seen as different folders. By clicking on the VMNs folder, the Markush structures are viewed in a grid view. The number of structures are indicated at the bottom left corner. You can select different structures by clicking on the arrows button on the top panel, or using the scrolling function on your mouse. Also, adjusting cell size is easy by resizing the width of the columns and the height of the rows. Changing the order of the columns can be done by dragging, which can be useful for different reporting needs. Same formatting and viewing options applies for the other tables. In this case, the example table, which has 9,477 entries. We have 3 different viewing options for the inventions table. For now, we are selecting the grid view. On the bottom panel, the number of entries in each table related to different views are indicated. You can select which records you want to work with, not only by using the arrow buttons and the scrolling function, but also by simply typing the row number of the molecule of interest in the text field between the arrows. Now we are selecting 19. By using the form view, a more detailed description for each record can be seen. Also, records from the relational tables are attached, such as the Markush structures from the VMNs files, the example structures, and also the details for inventions, which can make the life a lot easier by having the whole collection of info at a click of a button. More related data for the records can be added by using the Design tab, whereby widgets can be customized. Widgets can be removed, and also new widgets can be added to the view. The position and the size of each element are also fully customizable. So browsing molecules in our newly customized layout will provide us all the information we needed. Sorting records is also straightforward by right-clicking on the selected column and choose either descending or ascending. If you want to highlight cells whose data satisfies certain criteria, you can use Conditional Formatting option. For example, using the traffic lights option, you can highlight in green all the cells whose DCR numbers are not greater than a certain number, while the rest of the cells will be highlighted in red, for example. Also, exporting data into various different applications is easy, including exporting into Excel. You can specify details you wish to be exported into Excel by adding and removing fields. So the Excel spreadsheet has been populated with the data fields we selected, such as activity, description, and so on, for all the records we exported. Structure-based search is also possible by using the query builder, where you can add fields as structure. You can add the structure either by uploading from a file or sketching the query structure in Marvin. Running this query yielded 133 hits out of the 9,477 molecules contained in our database. More complex queries can also be made based on structure and other properties, such as activity, assignees, and so on. You can add more search fields to the query, and also you can remove them. You can select substructure or full structure search, or by selecting the ignore function, you can search for molecules that do not contain a certain structure. By clicking on the Markush structure, you can set the query features, such as the query atoms, atom list, atom properties, link atoms, the type of the bonds, whether it should be single, double, and or aromatic, and also whether it should be part of a ring or not. After setting up the query, we hit the Run button, and the hit list is returned in just a few seconds. Then one could select which R group should be shown. For example, the relevant R groups, all the R groups, and can also be set up for showing no R groups. Another important feature of IJC is the enumeration of Markush structures. The Markush and the query structure is shown in the two panels on the top. Three main enumeration options are available: full, random, and reduced enumeration, which is made according to the hit. Expanding homology groups is also possible, as well as limiting the maximum number of structures returned on the result list. Let's do a Markush reduction according to the hit. This option helps to decrease the complexity of Markush structures, so finding the query structure within the highly complex Markush structure becomes a bit more straightforward. If we click on one of the structures, we will see that homology groups are expanded. However, we can do enumeration with homology groups remaining unexpanded. Let's compare the two. The query structure is highlighted in blue, in which the homology group remained unexpanded here and is fully expanded on the other version. Also, by right-clicking on the Marvin viewer, one could select from various different options, including the different transformation options such as dragging, zooming, and 3D and 2D rotation. Finally, I will demonstrate full enumeration and the sequential enumeration of Markush structures. First, we start with a full enumeration and select display options. Also, we could use post-enumeration or chemical term filters before starting the enumeration, such as lead likeness, the Lipinski rules, and so on. But now we'll do an unfiltered enumeration. The output format can be also set, whether it should be screen, file, or database format. So let's run a full enumeration with unexpanded homology groups. The enumerated structures with unexpanded homology groups are shown. To compare these with expanded homology groups, I run another quick enumeration. We can see the difference between the two results here. To select which parts of the Markush structure should be further enumerated, we need to open our Markush structure in Marvin Sketch, whereby we can select the R group of interest. Then by clicking on Tools and then selecting Markush enumeration from the list, we can specify the enumeration option further, such as the enumeration of homology groups, the use of valence filter, and visualization of the results can be also customized. The list of enumerated structures are shown, of which we can select the one we want to enumerate further. A series of sequential enumeration can be performed by choosing an R group of interest at each step. This step-by-step enumeration can be applied as long as a single Markush structure feature exists. The sequential enumeration forms a fundamental part of the application called Markush Structure Interactive Browser, which is under development at ChemAxon.